So, announcements that you will care about. I've decided to move your test from Tuesday to Thursday. Still going to cover the same amount of material. Okay. You are going to have a take-home quiz over the weekend. And it's going to cover the nervous stuff that we already did, the brain stuff that we get through today, and all the way through the end of this set of notes so that I can get you to read ahead. Does that make sense? So I'll go in more detail over the stuff that I've already lectured over, less detail over the stuff that I haven't lectured over. Okay? So, and it will be due before you come to class on Tuesday. But actually before the 8:30 class on Tuesday, because that's when I get my first class. So, and I'll put that up on the announcement and everything when it's due. Does that make sense? Okay. So last time, FYI, 4 of you didn't do your take-home quiz, which is just hella dumb. Don't do that, right? A, it's free points and you should get 100. And B, I write it to prepare you for the test that is coming. And you need that preparation. So please do not do that again. And I will not ever let you take it late, because why the hell would I? I'm giving you like 5 days to do something that takes 20 minutes. You do not need to take it late. So the answer to that, so that you can save yourself an email the next time, is no. Because you had tons of time. Okay, so everybody clear? Okay, so we're going to do my very favorite stuff, so I'm totally nerd out today. Just get ready. If you thought it was nerdy before, it's getting worse, okay? So we're going to do brain today. Before we get into the brain notes specifically, I want to give you some understanding about how the brain is organized and how it develops. You ready? Okay. So the basic organization of the brain is that the very oldest portions of the brain, both evolutionarily and also developmentally, like the first portions of the brain to develop evolutionarily and developmentally, are the ones that are way down at the bottom. So like the brain stem is way down at the bottom, that's the oldest portion. And then the newest portions are all the way at the top and also superficial. Okay, does that make sense? So the brain develops both anatomically during development and also evolutionarily from the bottom to the top. The very, very last portion of the brain that you're developed is the most anterior, most superficial portion of your brain right here. Okay, so the big portions of the brain, there's 4 big sections. And so I want you to know that first. It's really easy, the brain, FYI, it's really easy to get lost in the trees and never see the forest. So let's start out with forest first, okay? There's 4 big regions of the brain. I want you to touch them as we go through. This is your cerebrum. Put your hands on your cerebrum. Because next time if you try to remember what the hell it is, you're going to say, hey, at least I remember touching it, okay? You have to do it. There you go. Okay, cerebrum. Ready? Okay, so the cerebrum is here. The cerebellum, right back here, just above your foramen magnum, which would be your cerebellum. Does that make sense? Your brainstem, stick your finger under your foramen magnum. That's about where your brainstem is. And you actually can't find the last portion, which is your diencephalon, unless you stick your finger up your nose. Don't do that. Just imagine it. Stick your finger up your nose and touch the bottom of your brain. So, in this picture, the only region that you definitely can't see is what? Diencephalon. The diencephalon is actually the region that the people have hardest time understanding, because you can't see it unless you slice the thing open, or unless you look from underneath and know exactly where to look for the diencephalon. Does that make sense? Okay, so what's this? Cerebrum. What's this? What's this? Brainstem. What's that? Diencephalon. Okay, so 4 big regions. The last one to develop is going to be the cerebrum, and the very, very last portion of the cerebrum to develop will be right here. Everybody okay with that? Okay, so we are going to start from the outside in. Okay, we're going to start from the cerebrum that you have had experience with and you know looks like a brain, and we're going to move in. Ready? So first thing about the cerebrum is that if you look at a top view, a dorsal view of the brain, you can see that basically there are 2 big halves, almost mirror images to one another of the cerebrum. They are called the cerebral hemispheres. Yes? Okay. Left and right cerebral hemispheres. What divides them from one another is the longitudinal fissure, also sometimes called the intercerebral fissure, but longitudinal is fine. This would run under what anatomical landmark in your skull? Sagittal suture. Good? Okay, got it. All right, so one more question. From last time, can anybody remember what chunk of dura would be in there? What chunk of dura mater would be in the longitudinal fissure? Go ahead. It is the dura mater, but there's a chunk that goes between. It's the meningeal layer for sure, but there's a chunk with a specific name that goes between the halves of the dura mater. Starts with an F. Falx cerebri, because you're in the cerebrum, right? So that goes between there. That was obviously removed, or else you couldn't see the longitudinal fissure. You with me? Okay. So the point I'm trying to make is this is all one story, not 72 separated stories. It's a little hard to put it together. I acknowledge that. And before we get into the thick of it, let me go ahead and tell you, just so that you can have an expectation. You know those things that you learn that you get the first time you hear them? The brain is not one of those things. You need to spend some time practicing, which is part of the reason that I moved your test back. So do not think that you are the only one that is going WTF as we're going through today. But you know that you did that with things that you now know, like the muscles of the damn forearm, right? What did you think when you first learned those? You're like, no way in hell am I ever going to know these. And then you did. Brain is the same way, okay? But it's super interesting. Well, I'm just super nerdy about it. I adore it more than anything, okay? So we have the cerebral hemispheres, left and right cerebral hemispheres. Notice that they're going to be really similar to one another. They're not exact, but they're pretty close to one another. They have a lot of similar wrinkling, and we'll talk about the similar wrinkling in just a second. So before we go any further, let's acknowledge that basically the primary characteristic of the cerebrum is that it has these folds, right, in it. And the folds have names. A hill, right here. This one, any of them, is called a gyrus. Plural is gyri. People say gyri, but it's wrong. And then a valley, yeah, is called a sulcus. Plural is sulci. People say sulci, but it's wrong. Okay, gyri and sulci. The point of the gyri and sulci are to give you more surface area without increasing the size of your head. Because let me tell you, the outer portion of the cerebrum, the cerebral cortex, is all of your high-level processing. It is all of your conscious awareness of sensation. It's all of your voluntary motor activity control. It's all of your intellectual processing and decision making, and the vast majority of your personality. So in order to be a human, you need a lot of surface area for that. Does that make sense? So how do you get a lot of surface area without increasing the size of the head? Yeah, you fold. So why not increase the size of the head? Well, I mean, we kind of understand that, but what happened during human evolution and primate evolution is at some point we did increase the size of our brain, and it actually is relatively big compared to our body size, but that's not the impressive part. The impressive part is the amount of cerebral cortex we have compared to our body size, the amount of folding on the surface. And what happened is our brain got bigger, but then it got to a limit because, of course, you need to get it out the vaginal canal. And there's a bone, right? There's a bony opening there. And so we had to basically do two things. A, we had to start folding more, right, with the same size of head. And then second, our babies have to come out when they're not done yet. They are half-baked, right? Who's mom or dad in here? Anybody? Okay, so let's just acknowledge to those the rest of the people who haven't had kids. When babies come out, is their functionality anything similar to what an adult can do? No, they're hardly even recognizable as the same species, right? They can't do a damn thing. They don't even know where their own damn hand is half the time. It's like a matter of weeks before they can put their hand in their mouth a lot of times. So they're half-baked. They need a tremendous amount of maternal care. They're not like a horse. Horse is born, horse gets up, horse is around, right? So they need to come out half-baked. And their brain continues to grow in size and surface till they're about 2.5 years old. when literally they start to behave like a human, right? 2.5 years, maybe 3, right? 6 months, my husband, he's a great dad, adore him, could not care less about babies until at least 6 months when they lock eyes with you. Might giggle when you do something. No, he takes great care of them. There's just like no like emotional attachment to speak of, just protection. Just like that's my thing, man. But later on, when he's super protective, like his oxytocin eventually kicks in too. But later on, when they're about 3 and you start being able to riff with them, like they've got a like serious bond after that. And now those two play punk rock music all day long and play video games together. So it's literally like his best little friend that just happens to be short. So they're born when they're really not done yet because they have to be. If you let their head get big, the woman wouldn't be able to walk away from the saber-toothed tiger, right? Or run away from the saber-toothed tiger. So this is the process of development. Now let me show you something to reinforce that. So during the process of development, this is an adult brain. Anybody have any idea what that is? That's a 24-week fetal brain. Just to let you know exactly how much is going on during that 3rd trimester, right? That is a really, really smooth brain. There is not a whole hell of a lot of capacity for intellectual processing going on right there. Do with that what you will. I don't want to argue with you. Okay, so now, in addition to the sulci, and we'll come back to that in just a second, in addition to the sulci, what we also have is these fissures. What's the difference between a fissure and a sulcus? Well, if a gyrus is a hill and a sulcus is a valley, a fissure is the Grand Canyon. And you don't have very many of those, right? You got a big one here and you got a big one here, right? And fissures, of course, let's put this back together. A sulcus would have which of the meninges in it? Just guess, it's fine. I don't expect you to know, but guess. What?
The shiny one.
The shiny one, good. What's the shiny one? Pia mater, and maybe a little bit of arachnoid, right? But definitely pia mater. But the fissure would have which of the meninges in it? All of them, right? The dura, the arachnoid, and the pia. Does that make sense? Okay. So those are the fissures. You also have your ventricles, which we'll talk about again. We talked about the lateral ventricles before. Those are within the halves of the cerebrum. And we'll come back to those in just a minute. But that's where your lateral ventricles are. Okay, so I want to talk about the cerebral cortex. Okay, so whenever you have a cortex of anything, whether it's an ovary or a kidney or a brain, it's the layer on the superficial surface, right? So the cerebral cortex, we'll look at it in this picture, then we'll look at a fresh brain. You ready? So this is the superficial cerebral cortex that you see. It is processing power, so you guys tell me. Should it be made of axons or neuron cell bodies? What does the processing? Axons are wiring. The processing is the cells, right? So that's why it's gray matter. It's actually processing power, okay? So let's look at an actual fresh brain, because it's actually not gray when you're alive. It's gray when you're dead, but when you're alive or just recently alive, it is kind of pink. Do you see that cerebral cortex right there? Okay, so this would be processing power, and this is wiring to and from the processing power. Does that make sense? Okay, so the cerebral cortex is, this is the very newest portion of the human brain, right? So everything develops from brainstem on up, so the newest portion. And it's the conscious mind. And let's look at some different mammalian brains to see what we have in common with our cat or our dog or whatever. So these are different mammalian brains, a bunch of them. And I want to talk about what the cerebral cortex is and how research supports what's different between you and your cat and your dog. So we've got a mouse all the way down here, and we've got a goat, we've got a giraffe and a human all the way up there, and then a chimpanzee. So what we tend to think, because it's easy to think, is that it's the size of the brain that determines intelligence, but that's actually not true. It's actually, a bigger brain is slightly better, but really what's better is a bigger cerebral cortex, yes, for intelligence, with reference to body size. So I want to put that together for you guys. Notice that a human brain and a giraffe brain are really, really similar in size, okay? But here's what the cerebral cortex does, among other things. First thing it has to take care of is it has to do conscious awareness of sensation, and you have to devote some real estate to the conscious awareness of sensation. And my body versus a giraffe's body, there's a lot more conscious awareness of sensation, real estate needed for a giraffe's body. Does that make sense to you? Right? Okay. But then the second thing it has to do is control voluntary motor activity of all of my skeletal muscle. Now I've got a lot less skeletal muscle than a giraffe. Yes? Okay. And then what's left you can use for intellectual processing, for personality, if you have the mechanics for language production, for decision making, for calculus, for algebra, for voting for Trump, whatever the hell you want to use it for. So you can use it for all of that later, right? So really what the big thing is the amount of cerebral cortex relative to body size. Do you see what humans have that is rare, rare, rare in other species, right? We've got a tremendously folded cerebral cortex compared to our body size, yes? Okay, so we are not the only species with that. We're the only species that also has opposable thumbs, hinged jaws, right? And some other things that we would need in order to manipulate that. We're also the only species that has developed a language that we can understand, right? Okay, so this is humans relative to, here's your dog and your cat. So if the cerebral cortex is the conscious mind, when does consciousness arise? Well, they actually call that in neuroscience, they call it the hard problem. Because does your dog have consciousness? Does your dog have some level of awareness, self-awareness? Is it the same as your 3-year-old? But is it probably a spectrum between mammals to a child to an adult? The levels of consciousness that you have, the levels of self-awareness, right? Does that make sense? So they call it the hard problem. When and where does consciousness arrive? When it's probably actually a sliding scale, right? Does that make sense? It's probably a sliding scale of like how much consciousness. It's not like a switch. It's like a sliding scale. And as far as we're concerned, we have the most complex level of consciousness, but probably because we can't talk to dolphins. Maybe they're like writing sonatas from from birth, and we're not. I don't know. So conscious sensory perception, this is where your conscious mind is. This is where your voluntary initiation of movements initiates. This is where your higher thought functions. And I am not saying that they are noble thought functions, but the fact that you can figure out how much change you should get, right? And argue with somebody on Facebook, and read an article and make those symbols into an idea, that's all in your cerebral cortex. Does that make sense? That's all where that is. So there's a ton of research about cerebral cortex, and there's a ton of research about the brain. But let me tell you a little bit about the history of understanding the function of the brain, just so that we can acknowledge this before we go. So one of the problems with understanding, especially the higher functions in the brain, is that we really don't have a great model organism to use for experimentation. So for instance, how could we tell if whatever our drug did relieved mouse depression? Right? Don't know. I don't know what a mouse looks like when it's depressed. It still looks like a mouse, right? So you can get up into the higher primates, but of course we can't study language to much degree in the higher primates. We have a hard time studying emotion in even the higher primates. So where did we get the information that we got? Well, some of it was anecdotal, like somebody gets a big old piece of steel stuck up in his prefrontal cortex, and all of a sudden Phineas Gage, when he wants to swear, which happens really deep, does it, right? When he looks at a boob and he thinks about it, he just grabs the boob. He just does it, right? His prefrontal cortex is not working, just boob grabber. Just think about that. Cortex, just telling you. So when you get this anecdotal information, if somebody gets a fork stuck here and they lose the ability to lick their left nostril, or a tumor over here and they can't speak, or they speak in a monotone or whatever. So it's a bunch of anecdotal information, but then I have to acknowledge the crappy history of humanity and where we got the rest of the information. The crappy history of humanity is whenever you have people who are not in power and people who are in power. Sometimes they made poor human rights decisions that resulted in some research that we're still using today. So people who were incarcerated, we would experiment on them. People who were in mental institutions, prefrontal lobotomies. If you want to take away someone's will, you just destroy this little will part right up here, right? So Nazi Germany did a lot of it, a lot of this experimentation. And the United States has a pretty sketchy history of experimenting on slaves. So that's where a lot of it came from. We still use it, but that's where some of the information, it's a really fascinating story to look at where we got all of this information. Okay, so you guys can read about that research on the cerebral cortex. So here's what I'm going to do with the cerebral cortex. There's no way in hell that I can tell you all of what the cerebral cortex does. But what I want to do is, for each part, I want to give you something to hold on to that you might be able to remember later. So instead of 72 paragraphs about the frontal lobe, I'll give you a few things, realizing that I have to oversimplify. Does that make sense? Okay, so before we talk about the cerebral cortex in detail, let me show you one cool thing. Two cool things. Somebody last year developed this simulation in which they developed a material that folds in the way that the cerebral cortex folds. And they actually, isn't it cool? And they actually developed it to show how the cerebral cortex folding occurs. This is not a real brain, but it's got tissue and a development that's similar to a real brain, which is cool. So before we go any further, I want to show you guys, maybe for the first time, what the texture of a real brain is like, because the brains that you guys are going to deal with in lab are preserved. And the reason they're preserved is because that tissue starts to disintegrate almost instantaneously after you take it out. And we can't get you fresh brains because mammalian brains actually carry prion diseases. So the brains that you guys are holding are injected with straight formaldehyde because that's what you need to fix that brain. So always wear gloves when you're dealing with them. It's not toxic unless you drink it, but it will hurt your skin if you leave it on there. It's really, really basic and it'll hurt your skin. Does that make sense? Okay, so are the sheep brains. Don't be scared of them. Don't lick the science, right, ever. It's just a good general policy. And wear gloves when you're dealing with them. But they are fixed. So they're going to hold their shape as you hold them. And if you wanted to get into them, you might have to slice. But I want to show you what an unfixed brain looks like. This is Suzanne Stensis. She's at the University of Utah in the neuroanatomy department. And she's got an immediate post-mortem brain that she's going to show you so that you can see the texture of the brain. And some of these structures that she's talking about we'll learn as we go through. I don't know how to make it louder though.
Surface of the brain. And what you see are the peel vessels. This nice blush with the clear leptomeninges.
That's the pia and the
vessels running in between the arachnoid and the pia. The cerebral spinal fluid has leaked out through the cisterns, and so that subarachnoid space is no longer visible unless I move the brain, but it's very, very soft. Notice, it's totally squishy. It's the consistency, it's much softer than most of the meat you would see in a market. So if I were to pinch this, in either way, I could easily damage this with my thumb. In fact, neurosurgeons, when they are doing surgery, often just evacuate with a vacuum or suck out parts of the brain.
So isn't that interesting? So if you had a tumor, you would think that they would be using a scalpel, for instance, but they can actually use one of those little dental suction tubes that's specific for surgery, but it's actually like, we'll just suck out a little chunk of your brain. And I could take a fresh brain and I could just pinch off a chunk. So I think it's kind of like a really, really fine bag filled with really cold pudding, right? That's what it kind of feels like to me.
This string here has been placed around the basilar artery so it can be suspended in a bucket of formaldehyde in order to denature the protein and harden it. If we didn't, it would sit on the bottom of the bucket and the brain would become deformed, as you would see here. so that it would be compressed in this direction just from the weight of the brain. So that points out one of the purposes of the cerebral spinal fluid is to float the brain inside the calvarium and act as a cushion. The cerebellum is easily seen, the medulla, the pons. Here you can see the arachnoid spanning across the space here at the base of the brain. The optic nerves are very obvious, as are the olfactory nerves. Here are our temporal lobes, and here is the uncus on either side. The uncus, remember, is the most mesial part of the temporal lobe. The tentorium that fit in here has been removed, but we can see a slight groove right along here where the tentorium sat. So there is no evidence in this brain of any of the temporal lobe herniating medially over the edge of the tentorium. It's a beautiful specimen. Unfortunately, this cancer patient died of complications secondary to a cell transplant. Here you can see how soft and pliable the dura is, and we can look down between the hemispheres and see the corpus callosum. I can also see a bit of the anterior cerebral artery coming over the surface. So this is the interhemispheric fissure.
Or longitudinal.
If I turn the brain around and we look at the occipital lobe, we can see the top of the cerebellum. And here there is a very small amount of blood that was actually seen on MRI that the patient had before death. If I let the cerebellum hang down a little bit, you can see the superior surface of the cerebellum. Turn it back around. So our very delicate and vulnerable brain, with its two vertebral arteries on the medulla, its basilar artery on the pons, it's branching to the posterior cerebral artery right here where you have the cerebral peduncle, and here we have the interspace. Here is the interpeduncular cistern, and we can see the two mammillary bodies.
Okay, so she has, you can watch this whole video, it's kind of interesting, and then she has another one about the spinal cord from just immediate post-mortem patient. You don't generally get to see fresh brains, right? Unless, of course, you're doing neurosurgery or you had something tragic happen. So the brains that you guys will be dealing with, and FYI, we do have human brains in the lab, both in the cadavers and out of the cadavers, and yes, you totally can touch them, right? Just use gloves, right? And if they're, do you know that all of those things in the jars over on the shelves over there, you can totally touch those. I just want you to ask one of the lab instructors to get them out. And you can see a lot of things that you wouldn't see otherwise. And of course, the proportions of a sheep brain are different than the proportions of a human brain, so it's good to look at the humans. Okay, so that's Suzanne Stensis from the University of Utah has a neuroscience department, and they also have a neuroanatomy and neuroscience graduate program. So if one of you guys wants to be a brilliant researcher, that's totally what I would have done if I figured out that I loved neuroscience before I went into molecular biology. My husband keeps going, why don't you go back? I'm like, I would be like 85 by the time I'm done. So I just like read about it all the time. Okay, so let's start talking about your cerebral cortex. You ready? Okay, cerebral cortex could not possibly be more interesting to me. So here is what happens with the cerebral cortex. Let's look at a simple picture first. In order to map out the cerebral cortex, it's divided into lobes, which are roughly under the bones that you know, right? So this would be the frontal lobe. It goes back a little further than you expect it to. These are the parietal lobes, yeah? Occipital lobes, I'm sorry, temporal lobes right over my ears, and then occipital lobes right here. That make sense? Okay, there's only one occipital lobe. So, actually two halves. So we're gonna start from the front. We're basically starting from the last thing to develop and then moving all the way back, That make sense? Okay, so if we want to look at frontal lobe anatomy, let's figure out how to find it. You need to be able to find some of the gyri and sulci that you use as landmarks. You ready? Some of them are really easy to find. So do you see this guy right here? It actually is the only one that really looks like it's going complete from left to right. That's called the Central Sulcus, and it's actually the boundary between your frontal lobe and your parietal lobes. Does that make sense? Okay. Now, if we look at it from the side, it's actually not hard to find, guys, even on a fresh brain. Do you see this one? Here it is. Right. That's not a fresh brain, but a preserved brain. That is the Central Sulcus. Okay. If you look here, this one is color-coded for you. So the frontal lobe would be everything anterior to the Central Sulcus, right? And everything superior to this sulcus you haven't learned yet. But this one's not hard. This one actually separates the temporal lobes over here over your ears. This one's called the Lateral Sulcus. Okay? And you're going to have to learn them all in lab anyway, so this is just your first time going through them. So the frontal lobe is all up here. That make sense? So we're going to learn just 3 little parts of the frontal lobe. It does a hell of a lot more than I'm going to tell you, but 3 little parts of the frontal lobe. You ready? Okay, the first part is a landmark that you can see anatomically and you can start to understand functionally. Do you see this gyrus that is just anterior to the central sulcus right there? That's called the precentral gyrus. Good? Precentral gyrus? Okay. Its function, though, we need to learn its function. You ready? Its function, and we're going to do that one first, right here. It is called, and listen to this stuff, make sure you try to let it get in, because you're not going to absorb it all right now, so you can write all you want, you're going to have to learn it later. So this, the function of this one, so the structure is called the precentral gyrus, the function is the primary motor cortex. Okay, so here's what it does, you ready? So this portion right here, just anterior to my central sulcus on this side, is controlling motor activity for skeletal muscle on this side. Everything that's going on over here, it's controlled over there. That make sense? Okay. The primary motor cortex on this side will control the opposite side. Does that make sense? Okay. So is this somatic motor or autonomic motor? Somatic motor, yeah. And it's voluntary control, right? So when I am doing that, I am controlling it voluntarily. Okay. So to reinforce this, not all voluntary motor activity is as tightly controlled as all others. So for instance, if I had to devote real estate on that strip to controlling my facial muscle activity versus the muscle activity of my gluteus maximus, which one would get more real estate? Facial muscles, right? Even though the gluteus maximus is bigger, it ain't writing any poetry, right? That make sense? Okay, so what happens is they have actually mapped out how much of that precentral gyrus is devoted to controlling different areas. You ready? Okay, so what you're going to look at here, this is a coronal section, right? And this is the primary motor cortex. You with me? They're showing it on this side, but it's actually this side. You can tell it's over on that side, right? They just pointed it to the wrong place. That makes sense? So you did a coronal section right here, and then they're looking at how much real estate you devote to, for instance, your hip. Not a whole hell of a lot of motor activity that you can do with your hip. Maybe Shakira's got a really big one. I don't know. But toes. No, not much. Look at your hand. How much real estate do you devote to motor activity of your hand? How much to your face? Yeah? Okay, so this is called the primary motor cortex. And tell me where it is again in the brain? Precentral gyrus of the blank lobe. The frontal lobe. Got it? Okay, precentral gyrus. So precentral gyrus is the frontal lobe. Okay, now, some brilliant person decided to take that, listen, and make it into a sculpture, yes, that is the proportions of the human body based on how much brain space they take for motor activity. I totally want this. You guys can get it for me, okay? And this is called the Motor Homunculus. Is that amazing? This is based on how much brain space is devoted to controlling motor activity of that. This is in a museum in somewhere in Europe. I can't remember, but a couple of people have actually replicated it. This is pretty freaking cool. Okay, so that's called the Motor Homunculus. Ready? All right. So, any questions? Everybody good? Okay. So there's two more portions of the frontal lobe that I want you to understand functionally. The first one is the prefrontal cortex. Okay, it's not shown on this picture for reasons I do not understand. So here's what the prefrontal cortex is. It's the most anterior, most superior portion of your brain. It's like right here. But only like the most superficial portion. It's the very, very last portion of your brain to develop because your brain develops back to front. This portion right here doesn't continue or doesn't complete development until you're around 25. So in some of you guys, you are not done yet. You're still baking, man, right? Okay, so it doesn't develop until then. So let's talk about this. It's really freaking interesting to me. It's got some real relevance right now. The prefrontal cortex, what it does is it is actually understanding the long-term implications of decisions that you make today. Yeah? So what is going to be the long-term implication of, I think I should take crack. You don't make as many of those decisions for the first time in your 30s, do you? I think I should sleep with this incredibly inappropriate person that I don't even like hanging out with. That happens before age 25 primarily, right? Okay. I think that I should join the military. Do as many people do that after age 25? They do not. I think I should get married because I'm going to love this person forever and ever. I think I should join a religion. Woo. You don't even understand the long-term implications of your decisions. Now, let's talk about whether that's absolutely bad to do. Is that risk-taking, poorly considered decision-making, an evolutionarily stable strategy for the species? Does somebody need to leave home and jump off a cliff to see if there's something better at the bottom of the cliff? Yes. Will some of the people die in the process? Yes. But will you probably potentially at some point find better, more rich places to live, right? Might you find somebody who's slightly more appropriate and you're more sexually attracted to if you leave your neighborhood? Yeah? But might you die in the process or make some others, right? So this is basically your teenage years through age 25. And you are making very, very risky but potentially very beneficial decisions from the time your hormones hit until the time your brain gets done, right? You make some interesting decisions. You also are incredibly idealistic. Look at the Parkland teenagers. And they can make big social changes. You write the best music then, you make the best art then, right? So we can diss them all we want for eating Tide Pods, but punk rock did not come from 45-year-olds. That's all I'm telling you, okay? It came from people who were in those years in which their prefrontal cortex had not developed yet. So there's really interesting things that happen in adolescence, and some of them later on you are going to be too conservative with risk in order to do. So that actually, and of course, separates a little bit with the sexes, okay? With the sexes, there's a couple of different things that happen. First off, which sex do you think is later to complete their prefrontal cortex development? Males, right? And there's an evolutionarily stable reason for that as well. Females, once they get into their childbearing years, especially if they have children, they need to be careful about their risk-taking decisions. Males, however, need to go find some new genetics. They need to leave the region where their sisters and their first cousins are, right? They need to do some risk-taking behaviors. They need to just try it out and see if it works, right? They need to be slightly more risk-taking. So the combination of the later prefrontal cortex development and the increase in testosterone, which increases your likelihood for risk-taking, is an evolutionarily stable strategy that will drive your parents crazy. That's all I have to say to you, right? You're like, what do you want the species to do? Absolutely, take some risks and find some new stuff. What do you want your son to do? You stay here. You stay right here, and she is inappropriate for you. and you do not smoke that, and you do not sleep with that person, right? But as a species, we would have still all been in Africa. 7 billion of us would have been in Africa if that had been the case. So my favorite neuroscientist is Robert Sapolsky. He wrote a beautiful, beautiful article about the benefits and disadvantages of the prefrontal cortex taking so long to develop. And if you guys ever read long-form journalism, it's a popular article. It's not like a journal article, but I'll post it for you if you want to read it. It's fascinating, especially for those of us who have kids. Okay, so that's your prefrontal cortex. Planning, reasoning, judgment, personality, intelligence, social skills. Social skills. So that dude, Phineas Gage, that got something stuck in there, right, in his prefrontal cortex, he forgot that it was really important to take the emotional drives that happen in your old alligator brain and layer over them social expectations. So he would grab a boob when he thought about grabbing a boob. He would swear at damn near everyone. I am vaguely suspicious that there are people in power in the government with poorly developed prefrontal cortexes. That's all I'm saying. Because they grab things and then they talk about it on tape, right? I don't even know. Would you? I don't know. Okay. So you can delete that editorial comment. Okay, I did not say names. The primary motor cortex is next. You ready? Okay, so the primary motor cortex, you ready? So here we go. Oh wait, we already did the primary motor cortex. The last part we're going to do is this part right here. Do you see it? It's just a little bit anterior to the primary motor cortex, right? This part right here. It's just this little blob, and it's only on the left side in 80% of us, and 20% of us it's on the right side, but it's unilateral on everybody that I know of, okay? This area right here is called Broca's area, or the motor speech area. You ready? I'm going to show you what Broca's area does. Here's what Broca's area does. What Broca's area does is, it is the reason my Broca's area is functioning, because I can look at this thing, and I can come up with the word water bottle, and I can produce the motor speech to say water bottle, or I can produce the motor activity to write water bottle. So not only do I come up with the right idea, it's putting the motor together to produce whatever symbology represents it, right? Does that make sense? So that's Broca's area. So when Broca's area has a tumor or stroke or lesion, what happens is I understand that this is a water bottle, and if you ask me for a water bottle, I could hand it to you. Yes? But you can't say it or write it. Okay? Now, sometimes you can say it, sometimes you can write it, sometimes you can't do either. It depends on how much of Broca's area is damaged. So I want to show you a stroke patient from a speech therapy clinic that has Broca's aphasia. Aphasia is a speech disturbance, right? Broca's aphasia would cause what? So what do you think would happen if I had Broca's aphasia and she asked me verbal questions? If she asked me what this was? I would know what it was, but I would have a lot of trouble getting out the words. Sometimes I might eventually get them out, but it would not be instantaneous.
So frustrating.
So oh my gosh, so frustrating. So it causes what we call halting aphasia, right? In which your speech is like interrupted because you know what you want to say and you know, right, that you're not doing it correctly. So I'll show you. This is Sarah. Sarah is, oh, not that one. You stay. Don't start playing. Okay, so this is Sarah. She's 19. And
okay. So what's your name?
Scott. Oh, no. Sarah Scott.
That's right. And how old are you?
I can't.
Try.
I can't.
on her right arm.
And also arm as well.
Leg.
Yeah. Yeah.
And what happened after the stroke with your speech?
Well, I can't.
You have speech problems.
Yeah.
Do you know what that's called?
Yeah. Aphasia.
Yeah, good, good. I haven't heard you say the word before.
I write it down.
And that helps.
Yeah.
Okay. So you were going to go to university. What are you doing now?
But
What do you do now? Every day.
speech, like,
Okay, so that's Sarah. Can you see what happened with her? That she completely understands the conversation that she's supposed to be having, but can't quite produce the motor activity dependably to have the output. Okay, so by the way, you can watch more of Sarah later. This was right after the stroke. A few years later, you can see how much functionality she's regained. She's regained quite a bit of functionality. Okay, so that's Broca's area. So you have 3 areas that you need to know in the frontal lobe, right? The precentral gyrus that does what? Primary motor cortex, right? The prefrontal cortex that does what? Planning, personality, yeah. And then Broca's area on the left side that does what? Motor speech. Got it. Okay, good. All right, so now we are ready to move back to parietal lobes. Okay, so the parietal lobes are here, so let's find the boundaries. Ready? So the parietal lobe starts behind the central sulcus, yeah? The lateral sulcus is below it, right? Or lateral to it. And then this guy right here is called the parieto-occipital sulcus. This sulcus is really, really hard to find, so let me tell you how to find it. When you're in the lab, if you have a brain or a model, look for it medial and then follow it out. Don't look for it lateral and follow it in. It's really easy to see on the medial side, but it's super hard to see on the lateral side. So if you find it medial and follow it out, you'll be able to find it. Okay, so that's called the parieto-occipital sulcus. Okay, so you're only going to learn one function of the parietal lobe. There are more. Okay, so the parietal lobe has this gyrus right here that's just behind the central sulcus, and so not surprisingly, it's called the post-central gyrus. Yeah, post-central gyrus. Okay, so, and the post-central gyrus is, listen, it is the somatosensory cortex. So, what kind of sensation? Somatic, from skin, muscles, and bones. From the opposite side, okay? So now we are going to have conscious awareness of the sensations that are going on in my skin, muscle, and bone. That means that me feeling this can be mapped to a specific region, the post-central gyrus, yes, on the right side of my brain. That's what somatosensory cortex means, right? It's the portion of the cortex involved in somatic sensation. Okay? Okay. So they've mapped it out just like they have in the other, the motor one. You ready? So here's the mapping for sensation. Watch. So do you think all areas of your body get the same amount of real estate for processing sensation? No, right? So think about this. If somebody touches your face, you can tell exactly where they're touching, right? You got a bug on your face, everything stops until you get the damn bug off your face, right? But if you have an itch on your own back, can you actually tell where it is? No, you're like, what? It's like, you know, you don't even know. Like, you have to do like process of elimination because you don't have a whole lot of real estate devoted to like that area, right? Does that make sense? So look at how much real estate is devoted to each area. So you got a whole bunch of real estate, sensory processing real estate devoted to your hands, a whole bunch to your face, yeah? Decent amount to your tongue compared to the size of the tongue. Look how much your elbow gets. You don't know what the hell's going on with your elbow. You're like, I hope it's still there, right? And then the big difference is that you got some real estate devoted to your genitals, yeah? Otherwise, there would not be 7.5 billion people on this planet, right? Okay? So, of course, some brilliant artist decided to make one of these for sensory as well. So now what we're going to take is we're going to take the portions of the somatosensory cortex and make them into a person that is proportional. All of the parts are proportional to how much real estate are devoted to them. It's pretty easy to tell the motor one from the sensory one. Anybody want to tell me why?
Penis.
Yeah, because you got some junk, right? When this one is the clitoris, it looks even weirder. It's like, I have literally never seen it that insistent. Because the clitoris has the same amount of real estate as the penis. So you just like make it huge. Okay, so this is called the somatosensory homunculus. This is how much real estate proportionally you devote in your brain to sensation. And I want this one too. And my husband is desperately afraid that I'm going to get them because he thinks I'm going to put them on the mantle, which I will. I totally will. So somebody is actually making them. I keep hoping that one of my lab aides, they have to do a project every semester. I'm like, if you will just make these for me. Well, go ahead. Hi.
There's a guy at Farmer's Market that makes like weird sculptures of that.
Really? Okay, I'll go do it. And by the way, the original ones are at a museum, but the idea is not original. Like, they've been drawing them for a while. So it's not like you would be really stealing somebody's idea. You're just basically taking a 2D idea that's been in the textbooks for generations and making it into a 3D manifestation of it. So, all right. So I love those things. They make me very happy. So the somatosensory cortex, right?
Oh, I'd be so pissed. I was like, I got your wife this beautiful gift.
He wouldn't, he's hardly surprised at all anymore. We have a poster from the 1940s in Germany above our bed that's like basically like reproduction. It's like all hand drawn and it's basically reproduction. He's like, I don't know if this is sexy. I'm like, just look that way. You don't have to worry about it. He's got musical instruments all over my damn house. I have to have my part. Okay. So the occipital lobes, we're going to move back to the occipital lobes. So the occipital lobes are easy because I'm not going to make you learn very much detail about them. And also we know a ton about them because what do they do? Yeah, it's vision. And actually we have a lot of model organisms that we can use for vision, right? Because we can see if we can figure out whether a rabbit's vision is destroyed, right? Yeah. We don't know what their personality did, but with vision we can. So this is vision. So it's back there, and basically the boundary that you're using is the parietal occipital sulcus and back. Now there's different areas of vision. There's some that's just awareness and there's some that's processing, but just call it vision. Please note that as much as I can, I've actually given you, if you can't remember a damn thing about the brain, but you don't want to flunk it all together, then I put something in caps for you for each region, right? That's probably not enough information to get you an A, but it's probably enough information to keep you from flunking, right? That makes sense? So if you want to like make an A, you learn a little bit more deeply. But I don't want you to leave anatomy with like no concept of what the cerebrum does at all. I want you to leave with at least a word for each region, right? Okay, and then let's move over to temporal lobes. You ready? Temporal lobes are super easy to find here, but look at like an actual brain. Can't you see the temporal lobe pretty easily? So this big sulcus right there makes it sort of hang off like a puppy dog ear. Do you see it? So that's the temporal lobe, and they're right here above your ears, right? That's temporal lobes. So if you look at the temporal lobes here, they do a couple of different things. They do, in both sides, the temporal lobes do hearing and smell. So hearing is easy to remember because they're right by your ears. But in the left side, they also do a lot of language, okay? In the left side, in 80% of us, they do a lot of language. And again, in 20% of us, it's on the other side. But it's unilateral, right? It's one side. Generally speaking, almost everything that goes on with language is on the left side, okay? So if you have a left side stroke, you're generally going to have some language impairment, okay? All right, so the temporal lobes are kind of beneath the temporal bone right here. The superior border is the lateral sulcus. They do hearing and smell, but then in addition, on the left side, they also are responsible for, and listen to this, because this is different than motor speech. This is word retrieval and language processing. So here's the difference. If we were talking about Broca's area, then if she asks me for a water bottle, I know what she's asking for and I can give it to her. I might not be able to write down water bottle or say water bottle, but I totally understand the meaning behind the words, right? You could see that Sarah was actually understanding what was going on in the conversation. This area is imparting the meanings to the symbols, right? So I can talk and talk and talk, and you can ask me a question about a water bottle, and I might talk about a bus, right? I don't understand particularly what you're saying, although I might grasp pieces of it. And I don't understand whether I am saying what I think I'm saying, although every once in a while I might get the words right. So does that make sense? This is putting together the idea in your head with the symbol, yes? So for instance, with this situation, what I might know is that I might remember, because it's in a different part of my brain, that you can eat an apple and it's actually a food item. But what I could not do is figure out anything about the word apple, right? I couldn't actually come up with the word, couldn't write it, couldn't say it. Does that make sense to you? So this is in a particular area right there in the temporal lobe that's called Wernicke's area. Do you see this little area right here? Okay. Wernicke's area is kind of at the border of the parietal and the temporal lobe, but they actually consider it a temporal lobe structure. It's kind of like right at the back there, right? And I used to call it Wernicke's area until one of my German students said, it's not Wernicke's, Melanie, it's Wernicke's. And then I asked them to pronounce, what did I ask them to pronounce? Huevos rancheros. I was like, okay, so say huevos rancheros, and he couldn't say it. I was like, there, we all have our own skills. Okay, so acknowledging that I pronounce it like a Texan and not like somebody who speaks Spanish. But so this is Wernicke's area, and Wernicke found it. That's why it's called that. And Wernicke's area is that area that we were just talking about. Wernicke's area is speech comprehension, both understanding what they're saying and understanding what you're saying. If you actually get a lesion or a damage in that area, it changes your capacity to produce the appropriate word or respond to the appropriate word. It produces a different kind of aphasia. Aphasia, again, is just a speech disturbance. But what would you predict? What would you predict? If we were having a conversation and I had Wernicke's aphasia, what would I do when she asked me a question? It's called fluent aphasia, in which I just talk, but I don't actually say the right words. Does that make sense? Okay, so I'm going to show you Byron, who, this is from another speech pathologist. Byron is just damn near the most darling person you could ever imagine hearing talk. I adore this guy. And he actually had a stroke in Wernicke's area, and you can see how it actually impacts his speech. You ready? So he's got fluent aphasia, also called Wernicke's aphasia. And you can talk and talk.
Hi Byron, how are you?
I'm happy. Are you pretty? You look good.
What are you doing today?
We stayed with the water over here at the moment, talk with the people over them over there. There's island for them at the moment. They'll save in the moment. He'll water first and for him. With love for him.
So we're on a cruise and we're not
We will see water right here and they'll save their hands right there for them.
And what were we just doing with the iPad?
I hope the world lasts for you. You know he was a nice person before that. He's just so freaking, oh, go ahead.
So they can totally like comprehend what's
No.
Oh, they can't?
It's not as if you lose complete comprehension. You have little pieces of comprehension depending on how much damage you have in Wernicke's area. But she asked him about the iPad, and he answered something entirely different. It's because you've lost the capacity to take the symbols of language and put them into the ideas, right? He can't put the symbols together with the ideas. With Sarah, she had symbols and ideas together, but she couldn't produce the symbols. Does that make sense? So he has understanding problems in both regions. But apparently, he's playing golf and he's pretty damn happy about his life, and he wants the world to last for all of us. And I do too, right? So like Byron is just like, oh, he's just darling. Okay, so now, so the regions that you need to know of the temporal lobe are really just Wernicke's area, but Wernicke's area is just involved in speech, and then you also need to know that the whole temporal lobe is hearing and smell. Is that okay? And then the last area is one you don't do in lab, because until recently I didn't have a way to show it in lab, but I did get a model, but you can look at it if you want or ask where the insula is. Okay, so let me show you where the insula is. I'm going to show you two different ways. So on the surface of the cerebrum, you won't be able to see it unless you actually pull the temporal lobe away from the anterior portion of the frontal lobe right there. Do you see? And then you'll actually see deeper cerebral cortex. Or you could look at it this way. Do you see? That's the insula right there. Yeah? Okay, it's still cerebral cortex, it's just folded in. So let me tell you a little bit about the insula. Okay, so the insula is another lobe. It doesn't correspond to a bone, but it's another lobe. And it's insulated, or the inside. Okay, so the insula function is kind of interesting. The insula is body awareness. It's like your awareness of where your body is in space, your awareness that your hand is touching your leg, right? Your awareness that you are folding your arms, right? You can pay attention to any of that. It's all there. But then in addition, it's part of your memory. Memory's super complicated. This is just a little bit of part of it. Taste is there. And then something really interesting, a really interesting area of research has gone on with the insula. Feelings of disgust cause the insula to light up like crazy. So, for instance, if you see a cockroach running across your food, the insula will be like on an fMRI, right? Or if you see spoiled food, or you see poop, right? Okay, especially poop in unexpected places. Right? Why is there poop? We really need poop to be sequestered in order to have a society. Let's agree. Okay, so the insula lights up whenever you feel feelings of disgust. Well, some brilliant researcher figured out that it also lights up in feelings of disgust for other people. And they have been using, this is voluntary, nobody's being incarcerated in doing this. Some people are really interested in what their inherent biases are. We all have inherent biases, right? You know your grandpa's a racist, but you might not know that you also, right, have some inherent biases against maybe people with disabilities, people who are not the same color as you, people who are not the same socioeconomic status as you, right? People who are a different sex than you, people who are bisexual, homosexual, transgender. You might not know that you have these. Well, some people are interested in what their inherent biases are so that they can overcome their inherent biases, right? And so somebody developed a way to assess what your inherent biases are. For instance, they have used this in some relatively progressive police departments. You probably need to know what your biases are. And I know we think, no, I'm not biased at all. I'm literally the most egalitarian person on the planet. Like using my own assessment to assess me, sure I am, but I'm not, right? I like grew up in a way that other people didn't grow up. What they do is they do an fMRI or another assessment that actually shows how much blood flow is going to an area. And they will show you pictures that make your insula light up and don't, right? Show a picture of a birthday cake, show the next picture with cockroaches rolling across it, right? And your insula lights up, right? Show a picture of poof out of context, your insula lights up, right? Show a picture of your grandma, your insula doesn't light up. Show a picture of somebody who is a different race than you, see if your insula lights up. Show a picture of someone who looks to be someone who is transsexual, see if your insula lights up. Show a picture of somebody who is elderly, see if your insula lights up. Disabled, right? So see if your insula lights up. So what do you do next? Well, what they do next is if the person is interested in further training to get over their biases, they can choose to do further training to get over their biases. So it's kind of an interesting thing just based on what we learned about the insula. And this has actually literally only been known or able to be studied for about 3 or 4 years. So go ahead.
Is it like learned or genetic? They like decided, because you could like nature somewhere just sway to like not liking like a certain race or gender or whatever.
What do you guys think?
I think that
has to be
they
both.
were more vulnerable to like for you raised that way and like learn that way. If
Yeah.
they have like things for.
So that's an interesting question, and I doubt there's one answer to it, but I have a suspicion that it's probably more one way than the other. I suspect that if you are raised in a house that it has disabled people and people of all colors, that you're far less likely to have that inherent bias that lights up. So I would be really surprised if biases against people of the same species were genetic. They seem to be reinforced by experiences, behavior, those kinds of things. Now, bias against poop, that's like way evolutionarily old, because that stuff's basically poison to us. So you can learn your way out of the biases. For instance, the next part of the study was after you go through bias training, and bias training is really interesting. The basis of bias training is that if you individualize people, it's very difficult to be biased against them. If instead of seeing an African American male and thinking scary, somebody tells you that this is Reginald, he likes to play Minecraft with his grandson, he likes hot dogs, but only if they have sweet relish on them, right? He has worked at this job for a long time, and his wife wants him to walk on the treadmill 3 miles a day, but he just tells her he doesn't do it, right? Then all of a sudden you're like, oh, Reginald is an actual individual human. And so with more exposure training, it's very, very difficult to categorize people very simply, which is, I think, something really fascinating, especially for right now. Don't you think? Something really fascinating. So if you can put people in a box, then you don't have to think about them as individuals. If you think about them as individuals, it's very difficult to imagine a box that would encompass all of the parts of their individual personality. So anyway, that's the insula. Okay, damn it, stop shocking things, Melanie. So last tidbit about the cerebrum is, let's address this. Let's see what kind of truth there is to this. So cerebral hemispheres, the left one and the right one, have a little bit of specialization in which the right one has some functionality that the left one doesn't. But this is really easy to make into a meme or a simplified story, right? You guys have heard left brain versus right brain, right? So is there truth to that? Is it true? Or is it truthy? Does it have just enough truth in it to make it continue to be propagated, or is it actually true? So let's see what's actually true. So what is actually true is that in the vast majority of us, the left cerebral hemisphere has slightly different functionality than the right cerebral hemisphere. So let's see what's generally true. The left cerebral hemisphere is where most of your pattern skills are. Can we agree that both language and math are patterns? Yeah, patterns of putting things together in different ways, right? How about music? Does music have any pattern to it? Yeah, music is math. I mean, the understanding and writing music is math. There's a lot more to music because you can impart emotional content to music. Well, so it's at least part pattern, okay? So the other thing about your left cerebral hemisphere is it really, really likes to understand. It needs clarity. It wants to be able to predict what comes next in the pattern. So this is really important for you guys to understand. And one of the things about the left cerebral hemisphere is if what you are learning has holes in the story, your left cerebral hemisphere will try to figure out the most likely thing that will fill that hole, and it will lie to you to give you peace. So if, for instance, you had a conversation and you forgot the middle of the conversation, your left cerebral hemisphere will fill it in for you, right? And you will remember what that person said during a fight because it fits the narrative that makes sense to you. So it lies to you to make sense of the world so that you can make further decisions. It's like, I almost know what the pattern, I almost know the pattern, I almost know the pattern. Oh, I think that's what that goes there in that pattern. So it's a big fat liar. Remember that when you're fighting with your significant other or your parents. You didn't say that, they didn't say that, you don't know what the hell you're talking about. Or maybe you did. So the left cerebral hemisphere is the one that people always consider mathematicians and engineers and those kinds of things. People who like spreadsheets, people who like that kind of stuff. So I am relatively left-brained, but do I not have the right half of my brain? I do. So let's talk about what's on the right side. It looks way sexier, let's just acknowledge it. I've tried to change my personality 3,000 times. I'm just the same person over and over again. So what does the right cerebral hemisphere do to you? Oh, by the way, and of course the left cerebral hemisphere has language, which is also patterns, right? So you've got language, masculine. If you get a problem in the left cerebral hemisphere, you're probably going to have a language disturbance. What does the right cerebral hemisphere do? Well, it does a lot of things, but among them, it is visual spatial understanding, yeah? And also imparting emotional content to the patterns that you generated in the left side, or understanding the emotional content that comes from the patterns on the left side, right? So visual, spatial, creative ability, but visual creativity. You know, I don't know, all scientists have to be very creative. They have to see a pattern, see a pattern, see a pattern, figure out how you could create a difference in the pattern. So creativity is not just like typically artistic creativity. So in addition, in the right cerebral hemisphere, the other thing there is face recognition and expression recognition. So if I say to you, two different things, if I say to you, yeah, right, or yeah, right. So same language, right? Totally different meanings, yes? It's the right side that allows you to distinguish between the two of them. Does that make sense to you? It also allows you to have facial recognition. Go ahead.
So does this side also help with sarcasm?
Yes, totally.
Don't understand
Totally.
sarcasm.
Yes, totally don't understand sarcasm. And by the way, sarcasm, you don't usually understand when you're really young. It actually develops a little later. So facial expression recognition, generating emotional content of language, or understanding emotional content of language. I know that you guys have had teachers, and I have too, that are completely flat in their delivery, right? I don't think anything's wrong with their right side. They may just have never audio taped themselves before and realized that I'm putting myself to damn sleep. So if you have a problem over here, it can make a person speak in a monotone and impart no emotion to the speaking voice. Odor discrimination is over here too. Usually if somebody has a seizure or a stroke and they like have odor hallucinations, they know it's right side. Right? Does that make sense? So it's over on the right side. So generating emotional content of language. One more thing about autism. One of the things about autism, among other things, is that you can't do sarcasm with people on the spectrum. You cannot do yeah, right, and yeah, right, and really expect that to get in, right? It's not something that is understood there. So you could teach them the difference if you did both of them, but it doesn't happen inherently. And a lot of times people with autism do have a flat monotone delivery. Do you guys, anybody know anybody with autism or on the spectrum? Okay, so now it's a spectrum, right? But you can have a relatively flat delivery for most speaking, and a little trouble with like vagaries of the delivery. Okay, so last little bit. So does that mean that right brain people are creative and left brain people are this? Well, first of all, you are not right or left brained. You are both brained. But the thing that you are best at is also probably the thing that you reinforce with behavior and the thing that you get reinforced for with behavior. Does that make sense? So I tend to be very left-brained because I grew up in the sticks, and literally the only way that I could get the hell out of there was with my intellect, was with my processing skill, was with my capacity to communicate, to solve problems, to do those kinds of things. Does that mean that I probably couldn't have done that? Who knows? I might have been able to, but I was predisposed to this, and then I was reinforced for that, right? Which is actually relatively rare in women. Are women actually reinforced for that behavior as much as they are reinforced for understanding emotional content of language and those kinds of things? So we are not all left brain versus right brain. We are both brain, but we do have a preference. And if that preference is reinforced, then what it does is causes synaptic plasticity in the brain, and you tend to get better and better and better at that, right? So left brain versus right brain is only truthy. That's all it really is. It's got a little nugget of truth to it, but you're not either any more than I am, although I do know a person with half a brain, and she is literally left-brained. Like, literally left-brained. Go ahead.
Good question. I have a sister who's left-handed. She insists that because she's left-handed, her logic side does not work.
No, that is not true.
So
Neil deGrasse Tyson is left-handed. He's an astrophysicist. No, what it is he's an astrophysicist. Neil deGrasse Tyson, he's left-handed. He's an astrophysicist. So it's not true. It just means that basically in the right side of her brain, the motor control is better, and that's genetic, by the way. She just handedness is inherited. Yeah.
So all she's done is merged with one side.
But you can tell her she's more likely to be a serial killer. Just statistically.
Well, I remember that.
So maybe you shouldn't piss her off.